Good evening, everybody. Myself, S. Ramachandran, Associate Professor, Department of Pharmaceutical Chemistry and Analysis, School of Pharmaceutical Sciences, Vistas, Chennai, is going to present a seminar on the topic Energy-Rich Compounds. Organisms require energy for various activities like muscle contraction and other cellular movements, like the active transport and synthesis of macromolecules. All these processes are energetically very demanding and use chemical energy. Chemical compounds liberate energy by hydrolysis of some groups which are bound to them by high energy bonds. High energy phosphate compounds are not used for the long term energy storage. They are temporary forms of stored energy and are used to carry energy from one reaction to another. The type of high energy bonds are the phosphoanhydrides, the enol phosphatic bond, the acyl phosphatic bond, the guanidine phosphatic bond, the thioester bond. The most important bond is the phosphoanhydride bond because it is formed between two molecules of phosphoric acid. Such kinds of bonds are found in your ATP, adenosine triphosphate. In ATP, there are two high-energy diphosphate phosphoanhydride bonds. The third between phosphate and the ribose is not much energy-rich as it is a phosphate ester bond. ATP serves as immediate donor of free energy in most endergonic reactions. Apart from ATP, GTP, UTP, CTP are also used as energy sources for metabolism of saccharides and lipids respectively. So in this slide, we can see the different types of bonds, various high energy bonds. This is a classic ATP, adenosine triphosphate. You have the adenosine group, the ribose sugar, and the three phosphate groups attached together by the phosphoanhydride bond, and one phosphate attached by the phosphoester bond. What is the working of ATP? The phosphate groups are held to each other by high energy chemical bonds. Under certain conditions, the end phosphate can break away and the energy released to the energy hungry reactions that keep a cell alive. When the end phosphate is released, what is left is ADP, adenosine diphosphate. This change from tri to di is taking place constantly as ATP circulates throughout the cells. The recharging of ADP to ATP requires huge energy investment, and that energy comes from the food we eat. So you can find out the hydrolysis of ATP to ADP, which is an exergonic reaction, and the dehydration of ADP, endergonic reaction. This is present over here. And the dissociation, AMP and other one. ATP forms ADP 5, ADP plus AMP plus 5. Or alternatively, AMP and pi and two pi bonds are formed. So this is the classic representation of ATP. The flow of phosphoryl group from high energy phosphate donors via the ATP ADP system to low energy phosphate acceptors. The phosphorylated compounds others are the phosphoenol pyruvate, the 1,3-biphosphoglycerate, phosphocreatinine, ATP, ADP, AMP, and PPI, glucose 1-phosphate, fructose 6-phosphate, glucose 6-phosphate, NADH and NADH, and NAD+. These are my references which I use for my study. Thank you.